In this video, we take on translation. Quite a bit more challenging. This is what happens at the ribosome to get us from the messenger RNA message to a protein, using tRNA as a go-between. Let's not forget that what we're making are proteins, right? They have complex shapes like collagen and hemoglobin that you see here. And that all starts with the humble amino acid. Remember, there are 20 different types of amino acids. We put them together in these long chains, usually of a couple hundred amino acids. We can choose from 20 every time and string them together in these long chains. And those adjacent amino acids interact, as we see here. And if you look closely, you'll see these dotted lines all over the place here. And those are hydrogen bonds that form between usually the amino group of one amino acid and the carboxyl group of another nearby amino acid. And these can form things like alpha helices or pleated sheets, just these basic shapes of amino acids. Then we have further interactions, and these are mostly caused by interactions between these R groups. Remember, R groups are that part of the amino acid that is different, and so if we have two hydrophobic R groups on sort of these distant amino acids, one of them here and one of them down here, they may wish to interact. R groups like this may want to form some hydrogen bonding between them, or even ionic bonding here. All of this adds up to the complex shapes that we see in the final protein, whether that's this sort of helical, triple-stranded collagen protein, or folded up four protein complexes like hemoglobin that carries oxygen around our blood. In the process of translation, who's going to be there? I said it's complex, so we have messenger RNA. Remember, that was the result of transcription before this. That then goes into a ribosome, is joined there by transfer RNA, or tRNA. Each of the tRNAs have this site where they carry an amino acid, and so there's going to be amino acids attached to those tRNAs. And then finally, this molecule that should look familiar, it looks a lot like ATP, but this is guanosine triphosphate, and this is going to provide the energy that we need to string together the amino acids. Well, the process is called translation for a reason. We're going from one language to another. Essentially, this nucleic acid code language to a protein sequence. So we're switching chemical languages, and the key to that is transfer RNA, or tRNA. It is the key to the genetic code. I introduced it before. It is a long stretch of RNA, so single-stranded RNA, although it does have some interactions where it's bonded by hydrogen bonding and therefore double-stranded in a couple of spots. But really, a lot of times we just make it a bit of a cartoon. So here's our big tRNA molecule. The key parts of that are the anticodon that are complementary to the codons on the messenger RNA, and the amino acid binding site where it carries a single amino acid. So each tRNA, and there are 64 different types, carry their own amino acid. Just like transcription, we're going to have the same three phases: initiation, elongation, and termination. And like transcription, initiation is actually kind of the trickiest and most crucial part, for a slightly different reason though. So in transcription, we are trying to find where is the gene that I want to use out of all of these possible genes. In translation, I want to find the start codon. So on our messenger RNA here, in this case, I'm going to actually start at the 5' end, and I'm going to move down the messenger RNA until I find the start codon. That is always AUG. This establishes what we would call the reading frame. What do I mean by the reading frame? Well, remember, these codons are written in 3-letter sequences, kind of like this sentence down here. Now, it's just a string of letters, kind of like this messenger RNA is a string of nucleotide bases. A little tough to translate, but if you look at it and we put spaces in between each of those, it's pretty easy. Well, maybe a little gruesome, but it's pretty easy to interpret. If we start at the right place, if we started the every single time, it'd be kind of like if you started every single sentence you spoke with the word the, then we would know that we're beginning a sentence. But if you started the wrong spot, it becomes completely unintelligible, right? If we begin essentially here, then the sentence instead of the dog ate one cat reads Edo gat eon eka. Doesn't even sound like the same language. If we're trying to make a protein, this would make the wrong protein, this would make the right protein. So establishing the reading frame is really important. Always start at the start codon, and everything else kind of falls in place from there. Once we get a tRNA carrying its amino acid, always methionine, bonded to the start codon, then we use a little bit of energy and assemble the other part of the ribosome. Now we have all the players together. We have our transfer RNA carrying an amino acid, we have the whole ribosome, and we have our messenger RNA. We're ready to keep going and assemble the rest of the amino acid sequence. Elongation is fairly simple, kind of like in transcription. We just keep growing the thing that we're building. In this case, a chain of amino acids making up a polypeptide. And the rules for it are simple. They're our old friend complementary base pairing. Complementary base pairing is what is going to tell our transfer RNA that this is the spot for you to bind to. So we have on the messenger RNA a codon. We always started with that AUG codon. And so the transfer RNA here carries with it, as part of it, an anti-codon. And so if it has the complementary string of UAC, then we get complementary base pairing, and then we know for sure this is the right transfer RNA at this particular spot. So what that means essentially is that this tRNA can park in this parking space. Once it does, we spend some energy in the form of GTP, and that helps codon recognition. They're sitting there next to each other now. So these two amino acids, and so the amino acid on the left, and each of the different amino acids are represented by different purple shapes here. And there's this chain here on the left, and it's sitting right next to a new amino acid. We're going to form a peptide bond. And so here now, at this next stage, we have a long string of amino acids in a growing polypeptide chain. And that just keeps getting longer and longer. Again, elongation. Well, now this tRNA essentially has nothing to do. It's given up its whole chain of amino acids, so it really needs to leave. And to do that, it needs to essentially move over into the next parking space. GTP, again, is going to be the energy we use to get the ribosome essentially to shift over. This tRNA can then leave, and this tRNA right here, which was the one that came in up here initially, right? It carried this diamond-shaped amino acid, and there's that diamond-shaped amino acid. Well, it's now in this P location, and it's ready for another tRNA to come into this spot so it can bind another amino acid. And the process repeats itself in the cycle time and time again, bonding more and more amino acids into this longer and longer polypeptide chain until, of course, we get to the end. The ending in translation, again called termination. Similar to transcription, we hit a stop sequence and then everything falls apart. So in this case, there are several different codons that say stop, kind of like coding for a period at the end of a sentence. And so if any of these three codons are the ones that show up, then a different type of tRNA binds there. It's a release factor. This one does not carry an amino acid with it. It binds to this site and allows everything in this next step to be released. We release that tRNA. The whole polypeptide chain is now free. It's not bound to anything. And the ribosomes fall apart into their two subunits. This messenger RNA here can go find another ribosome and go make even more polypeptides. So basically, everything is freed. We have our polypeptide, which again was the goal. That's been achieved. We made a polypeptide. Now it needs to be used. In order to use that polypeptide, we need to fold it. So all we have right now is a string of amino acids. And so there are other proteins that help this process of forming these molecular interactions and finally getting us to this folded up useful shape of a polypeptide, often with several of them together, like in hemoglobin, where we have 4 different proteins assembled into one complex. And remember that in proteins, it's the shape that determines their function. So this is a really critical step. Now that protein is folded and it can be shipped off either within the cell or out of the cell to do its job. Let's make one more stop and put this into the context of the cell. So really difficult sometimes to get a grasp of what's going on at a molecular scale, but remember, we were just doing translation at these little ribosomes in here. And see all these dots all over the place? So those are lots and lots of ribosomes making proteins. If we're making them here in the cytoplasm, these free ribosomes, we're probably making proteins that are going to be making up the cytoskeleton or enzymes or something else that stays within the cell. But then we also have the rough endoplasmic reticulum. And so it got its instructions from the nucleus, right? We did transcription in the nucleus and we sent messenger RNA to the rough ER, which also contains those little dots, the ribosomes. And if we make it there, then we can pinch off a completed set of proteins in a vesicle, right? So we've got the vesicles, and they're going to transport those proteins to the whole Golgi body, which is going to chemically tag it and probably secrete more vesicles to come to the cell surface and either put the proteins in the membrane itself or to expel them out of the cell in the case of something like digestive enzymes that your stomach or intestines might produce. If you like, we can run through the whole translation process again using the paper models that we looked at in class. So I have my messenger RNA. It's out in the cytoplasm and it needs to find a ribosome. And so to start all this in place, I'm not going to actually show a ribosome. A little too difficult. It would be a very large protein that would come around it, actually a two-parted protein. But the first thing I need to have is some tRNA. So to get things started, and we call that initiation, just like in transcription. In translation, we need to start by getting a tRNA in place. It seems like this would be the appropriate one. This transfer RNA carrying glycine, it is complementary, again complementary base pairing to this. However, if I did this, remember that we're read in sets of 3 nucleotides called codons. If I did that, the rest of these amino acids would be completely wrong. To avoid that problem, we need a common starting point, and that is that we always start at the codon AUG, and and that always carries the amino acid methionine. So here's our transfer RNA carrying methionine. We have that linked up to the messenger RNA, and then the ribosome would assemble around it. Initiation done. So the rest of this, elongation, again driven by complementary base pairs. The ribosome helps all of this out. We need to find a tRNA with the complement to UAC, which would be AUG. So let's find one with AUG. Here it is over here. So the next one that comes in place is this transfer RNA carrying the amino acid tyrosine. Now that these two are together, we add some energy in the form of GTP, similar to ATP. That transfer RNA leaves, the ribosome moves over, and now we look for one with CCC. So there it is. These two are together, we form a peptide bond there. This one then can leave. Now we're looking for CAC. Here it is right here, carrying valine. These two are linked. That goes off. And now we need another CCC. I don't have one. I have a CCU. What actually happens to these? I mean, what do you think happens when this leaves? It's got this spot on the top of the tRNA. There's just two important parts, the anticodon and the amino acid binding site. So what this can go do is actually find another amino acid, glycine, carry it in here. This one leaves to find another valine and link up to it. These two have a bond. Now I'm looking for another one, AUU. And this is a unique one. This one does not carry an amino acid. This is our signal that we're done building the protein. Now normally this would be several hundred amino acids long perhaps. So we've just done a really short polypeptide, but this says we're done. And this is the termination site, the termination stage that allows my protein to go off and do its job somewhere. It's going to be folded up just right. tRNAs can leave, this can leave, the ribosome falls apart, and this messenger RNA can go find another ribosome, more tRNAs, and form more proteins. That's the process of translation.